So today's Monday, October 31st, and the point of this video is to inform those that were absent or quarantined what we did today. So if you go to the calendar for August, and then you find, of course, the date, so August 31st, it'll take you here to what we basically kind of covered in class. So let me kind of break this down for you so you can see what happened. So we did a cell CER poster presentation. So a handful of student groups put their posters up on the smartboard and presented their claim, evidence, reasoning for Euglena cell being plant, animal, or whatever it was. So then students submitted that assignment. So make sure that you have submitted that. That was due today, but I had them do it in class while that because it was already done. And then we talked about the cell lab report. So the cell lab report, so I'm going to jump back here and go home. And we are still currently in Module 1. And within Module 1, we are on the new week. So I'm going to scroll down to week 2. So cell lab report. So here's what I asked them to do. For those of you that have me in class before, or when you think of a science lab report, what you're about to see is not generally what we expect. So this does not follow the normal lab report format for science. We're kind of getting our feet wet with some things, and because of the way the procedure worked, this just works out really well for what we are doing. So you're going to basically write a report that includes 3 sections, and it's going to answer the following questions. So one, what question were you trying to answer? So that's basically your guiding question. And why would this type of question be important to a scientific community? So in biology, high school biology labs, we're not discovering anything new. We're not on the forefront of science. So we're doing labs that we basically already know the answer to. But I want us to think like we may be on the cusp of doing science. So with this particular lab, I want you to think about, like, what if this organism was an actual unknown organism or an unknown cell or whatever it was? How would we behave like that? So why would this type of question be important? So what would this help us do? The second thing is, what steps did you take during the investigation? I'm not looking for like step 1, step 2, step 3. I got a microscope, I looked through the thing, so forth and so forth. I'm looking at what observations were made and things like that. And I realized that you potentially were not in class to do this. So if you weren't, you're still kind of doing, because I don't want a step-by-step process. You're still doing what I'm asking for. You took data, you collected data, so forth. In that same section, why do you think this lab was conducted this way? So instead of just saying, why do we do the lab, which is the answer is like Mr. Hartman told me to, why would we do this if we were trying to really investigate an unknown organism? And then the last one is, what is your claim, evidence, and justification? So taking that poster that we just submitted and just went over and putting it into a paragraph form. So this is going to be a report, and I say report in the sense of what you would think of probably like middle school. The report should answer these questions in about 1 to 2 pages. I'm not looking for any grand scheme of anything. It's got to be typed. If you want to put diagrams or pictures in there, tables, you can. Be sure to write in a persuasive style. You're basically using this to convince somebody that the study, the research that you did was valid, and that we want to convince them of something. I would also recommend to, I guess recommend is a strong word, do not change your claim. At this point, I've told everybody what a euglena is. It is a protist, which is kind of in between a bacteria cell and a animal plant cell. It actually has properties of both, so you can look it up. But it does have animal parts and cell parts, and the reason for that is it wasn't a trick. your claim is going to be right, basically, no matter what you really stand on, because it has both animal, it has both plant. If you went on both, so all that's right. So that was the first part of class. Going back to the lesson, so we did that. We did a water phenomenon, which there's a video linked here. It says square water, which will kind of show you basically what we did. If you would like to do it at home, it's pretty simple. You just need wax paper, some water, toothpick. And so what I had them do was put the wax paper down, put some drops of little water on the wax paper. It beads up. You can kind of move around with the toothpick and kind of investigate that. Then I came back afterwards and gave them a toothpick with some soap on it. So just some Dawn dish soap. Put that on there and look what happened. The video kind of does the same thing, but just a lot different and kind of a bigger, cooler process. So if you watch the video, you'll kind of have that idea. And then I gave the property of water notes in class today. So we did that. I tried to record it as I was putting on the document camera. I don't think it works too well. I don't know what the problem is. I'm going to post that video as well right here where it says under square water and where this video is. So you can get that. Unfortunately, because I had to run out after third period and get out of way of another class, I left the completed notes at home, or not home, sorry, at school. I'm at home. So I will post those here if you can't, if the video really doesn't work. It blacks out for a minute. It seemed to lag there towards the end. So you might be able to kind of fish it out. I don't necessarily expect you to do that, so I'll post those. So that's basically what we did today in class. So I hope that helps. If you have any questions, please feel free to shoot me a remind, shoot me an email, and I will try to get back to you with as much as possible on what those are. Oh, speaking of the doodle notes, it isn't a Kami assignment, so that way you can type on them, fill them in. I do not expect you to turn those in. That is not the point of that. Anything through Kami has to be an assignment, so you can edit it, but it is not an assignment, so don't worry about turning that in. So as far as due dates go, I understand you are quarantined. Obviously, I'm not putting this together until currently at 7:20 tonight. So I'm lenient with due dates as far as this goes. We will work together. I'm more concerned about you learning content than strictly enforcing due dates and late work and penalties and things like that. So just do your best to put it all together. We've got to be adaptable here and work together. So I hope that helps, and I will see you in the next video.